Industrial wastewater contains various pollutants, including dyes and color compounds, which can negatively impact the aquatic ecosystem and water quality if discharged without proper treatment. Monitoring environmental quality parameters is essential to assess the level of contamination and ensure compliance with the environmental regulations. Spectrophotometric and colorimetric techniques are widely used for analyzing wastewater because they provide accurate, rapid, and reliable measurements of color intensity and dye concentration. These methods are based on the interaction between light and dissolved substances, where the amount of light absorbed is related to the concentration of the analyte. And therefore, environmental quality assessment plays a crucial role in protecting public health and maintaining sustainable industrial practices.
For experiment 1, the chemical we use including first reactive black 5, second distilled water, third is the industrial wastewater sample, and the fourth is the dye. And then the apparatus we use including beaker, volumetric flask, measuring cylinder, filter funnel, filter paper, tweezer, centrifuge tube, and then the micro pipette, the wash bottle, and also the Janeway colorimeter. So that's all for the apparatus and chemical used in experiment 1.
Now, I will explain the procedure of this experiment. According to laboratory manual, the experiment involves the determination of environmental quality parameters in industrial wastewater, including COD, ammoniacal, nitrogen, and dye concentration. However, due to unavailability of the DR6000 spectrophotometer during the laboratory session, only the dye concentration analysis was carried out using the Janeway calorimeter. First, the Janeway calorimeter was switched on and allowed to warm up for approximately 15 minutes to stabilize the instrument. A suitable wavelength was selected and distilled water was used as a blank solution. The blank was inserted into the calorimeter and the instrument was set to 0 before taking any measurements. Next, a stock dye solution of 1000 ppm was prepared. Several diluted dye solution with concentration of 5 ppm, 10 ppm, 50 ppm, 100 ppm, and 500 ppm were then prepared using distilled water. Each solution was mixed thoroughly to ensure a uniform concentration. After that, the transmittance of each standard solution was measured using the Janeway calorimeter. The industrial wastewater sample was also analyzed in the same manner. All readings were recorded and compared to observe the relationship between dye concentration and transmittance. The collected data were then used to evaluate the level of dye contamination in wastewater sample based on the Beer-Lambert law.
Based on the graph and table, the percentage transmittance of different sample was measured at various wavelengths ranging from 430 nm to 700 nm. The results showed that the 500 ppm sample has the highest transmittance value with a maximum of about 0.76 at 430 nm and generally remaining higher compared to the other concentration. This indicates that the sample allows more light to pass through as the concentration decreased from 500 ppm to 5 ppm. The transmittance value also decreased. The 100 ppm and 500 ppm sample show intermediate value, while the 10 ppm and 5 ppm sample have much lower transmittance close to the wastewater sample. The distilled water sample show almost zero transmittance, which indicates no significant absorption or interference from impurities. Meanwhile, the wastewater sample show very low transmittance values, suggesting the presence of suspended particle or dissolved substance that reduce light transmission. Overall, the trend showed that higher concentration sample produce high transmittance reading, and wavelength change also affect the light transmission. This analysis helped to compare the optical properties of the sample and evaluate the presence of contaminant.
Hi, my name is Kylie. So for the discussion part, describe the purpose of using the Janeway colorimeter. So a Janeway colorimeter was utilized to measure the absorbance of the dye solution at discrete wavelength. So the maximum absorbance was identified at 430 nanometer, and this allowed for the accurate determination of the unknown wastewater dye concentration by manually calculating the linear regression and demonstrating that single wavelength spectrophotometry is a viable alternative when automated system fail. And for the second question, compare ADMI method for color determination with other method. So ADMI measures total color difference across the entire visible spectrum using the Adams-Nickerson formula. It's ideal for complex, mixed industrial effluents where specific dye are unknown. And single wavelength measure absorbance at one specific peak. It's accurate for single known dyes but fail to account for visual pollution caused by mixed color. Moving on to the third question. Describe the mechanism of reaction of the COD reagent and ammoniacal nitrogen with sample by evaluating the changes of color, temperature, and chemicals reacted during the reaction. So for the COD, the thermal energy drives the oxidation of organic waste by potassium dichromate. As it oxidizes the waste, it reduces the chromic ion, shifting the color from orange to green. So for the ammoniacal nitrogen, the Nastle's reagent react with ammonia at room temperature to form a complex compound. So this produces a yellow or brown color whose intensity is directly proportional to the ammonia concentration. For the fourth question, relate the experiment to Tauhid paradigm. So for the practical, strictly transferring precise stock volumes to minimize the chemical waste and prevent stock contamination. And for the tolerance, dividing complex tasks under pressure to ensure accurate result despite equipment failures. And for the value, enforcing proper PPE usage, managing hazardous waste safety, and ethically reporting the equipment failure to calculate a valid alternative solution. For the last question, discuss the importance of environmental quality measurement in industry. So the importance is to ensure industrial facilities comply with legal discharge limits to prevent high COD effluents from depleting aquatic oxygen levels. Yeah, that's all from me.
So now we move on to the question part of the experiment. The first question is, why is it crucial to heat the COD vial? Heating the COD vial to around 150 degrees Celsius is important because it allows complete oxidation of organic pollutants by potassium dichromate. If the sample is not heated properly, the reaction may be incomplete, leading to inaccurate COD readings. Therefore, heating ensures reliable and accurate result. Next, for question 2 is, determine the regulation used for the experiment. So the answer is, this experiment is based on Malaysia's Environmental Quality Act 1974 and the Environmental Quality Industrial Effluent Regulations 2009. These regulations set the standards for industrial wastewater discharge and help control water pollution. Next, determine the minimum requirement or limit for wastewater according to regulation and its importance. According to the regulations, the COD limit is 120 milligrams per liter under standard A. For ammoniacal nitrogen, the limits are 10 milligrams per liter for standard A and 20 milligrams per liter for standard B. These limits are important because they help industries determine whether wastewater is safe to discharge and complies with environmental requirements. And the last question is importance of the industrial regulation and its impact on society and environment. Industrial regulations are important because they protect water resources and reduce environmental pollution. They help preserve aquatic ecosystems and ensure public health and safety. In addition, these regulations encourage industries to manage wastewater responsibly and support sustainable development. That's all from me. Thank you.
I'll take part from here and continue with the part of conclusion. My name is Oiwan Lee, my matrix number is AW240120. So although the wastewater samples indicate an equivalent dye concentration of less than 5 ppm, but this alone does not guarantee the compliance. This ADMI index requirement under DOE Standard B is stricter. It specified that the effluent must remain or below 100 ADMI units and exhibit the no objectional color. In the dataset, the filtrate still produced measurable absorbance across the several wavelengths, confirming that visible coloration persisted. This residue color means that the wastewater is not compliant and cannot be discharged without further treatments. To achieve regulatory standards, additional polishing steps such as activated carbon adsorption, advanced oxidation process, or biological degradation are required to fully eliminate dye molecules. Implementing these measurements ensures that environmental safety, prevents ecological harm, and reflects the Tawhid paradigm by integrating scientific rigor, ethical responsibility, and sustainable resource management in industrial practice. That's all from me. Thank you.